This lecture is going to cover tonicity of cells and how water is going to move in and out of those cells through osmosis. So before we get started on some new material, I want to make sure that we are remembering some terms that we've already covered this year. So the first, we have to talk about the solute. So that is what is being dissolved in the solution. So remember that there's a solution inside of the cell and there's a solution outside of the cell. The solvent is the substance that does the dissolving. So the majority of the time it's going to be that universal solvent, which is water. And the solution, so that's the solute and the solvent put together. So just keeping those terms in mind because it's going to be important as we are looking at the concentrations and how they vary inside and out of the cell. So remember that the solution that is inside of the cell is going to have its own concentration, and then the solution that is outside of the cell is also going to have its own concentration. So they may or may not be the same, and that's where we're going to have some movement of water occurring. So some other things to keep in mind is that cells are always trying to reach this magical point of equilibrium. So we're always trying to balance out the concentration inside and outside of the cell in terms of the solute. We're going to use transport for this, and then what we're going to do a lot of the times is move water through osmosis. It's a passive type of transport, so the cell doesn't have to use any of its own energy to reach that equilibrium. So what you want to remember is those solutions that we just talked about inside and out of the cell. We are going to be comparing those. So how does the solution outside of the cell compare to the concentration of the solute inside of the cell? So another kind of review here, just to get us thinking about these terms. So if I have an area that is hypertonic, that means that it has a higher concentration of a solute than the area that we're comparing to. So if my cell, let's say that my cell is hypertonic, that means my cell is hypertonic in comparison to, let's call it the environment that the cell is in, or the solution. What this also means is that your concentration of water inside of the cell is going to be lower than it is outside of the cell. And we'll do some practice problems to help kind of clear that up a little bit. But just keeping that in mind that we're talking about solute concentration, but there's also water in all of these solutions. In a hypotonic area, that means that area, whether it's the cell or the environment or the solution that the cell is in, it's going to have a lower concentration compared to the other. So either the inside of the cell compared to the outside, or the outside of the cell compared to the inside. In this situation, that area is going to have a higher concentration of water. And then if we are isotonic, it means that our concentration of solutes inside of the cell and outside of the cell is equal. All right, so some things to keep in mind before we look at some situations to see how water is going to move and what effect that's going to have on the cells. The first is that it's easier to move water than to move the solutes. So we could have things like glucose, which are generally too large to move without a protein. When we have things dissolved, a lot of the times they're going to dissolve into ions. So if something is charged, then we're going to require active transport. So we don't really want to move those things. We want to move water, which means that we're going to use osmosis. And then a key thing to remember is that osmosis is when water is going to move from an area of high concentration to low concentration. The key to keep in mind here, we're going to be talking about concentrations of the solute, we're going to be talking about concentrations of the water, and what's low and what's high and what's inside and what's outside. When you're not keeping track of it, it can be a little bit overwhelming. So what you want to think about with water is water is always going to move from where there is a high concentration of water to where there's a low concentration of water. Okay, so it's not just water is going to move from high to low. You have to think about, am I looking at the high concentration of water, or am I looking at a high concentration of the solute? So let's go ahead. Let's take a look at an example here. So as you're looking at my little drawing here, this circle right here, that's going to represent the cell. Okay, that number inside represents the concentration inside the cell. And then this area right here is the solution that the cell is in. Solution, sorry guys, of cell. So we're going to have to do a little bit of math, not too much, but what you want to remember is that the solute and the solvent, their percentages or their concentration will always equal 100 within the same area. So what do I mean by that? If 20% of my solution is made of solute inside of the cell, that means that, I'm going to do this in blue to represent water, that means that 80% of that solution inside is water. Okay? So outside of the cell, if 10% of my solution is solute, that means that 90% is water. So just some simple math there. So your total is always going to equal 100. Now, the first question we have to answer is this one I just circled in purple. The solution is blank. So is the solution. That's the one that the cell is in. If I was asking for the cell, it would say the cell. The solution is hypertonic, hypotonic, or isotonic. So we're comparing the solute outside to the solute inside. So this solution, the 10%, is less than 20%, which means that this solution is hypotonic. Okay, so that's step one. The next step is to identify in what direction will water move. So if you remember, what we said before is that water will move from a high concentration of water to a low concentration of water. So where's my higher concentration of water? Well, that's outside of the cell here. I have a higher concentration outside, 90% to 80% inside. So what's going to happen is water is going to move into the cell, causing the cell to reach equilibrium. So water will move into the cell. So let's take a look at another example here. So same thing, remember this is my cell. This area here represents the solution that the cell is in. Okay, so you can do it one of two ways. You can start with this question here, or you can start with your concentrations. Since we did our concentrations first last time, we'll do it again this time. So inside myself, 10% is solute. That means that 90% is water. If then outside the cell, 20% is solute, that means that 80% is water. Now, when determining this solution, if it's hypertonic, hypotonic, or isotonic, we're going to compare the outside to the inside. So the outside in this situation is 20, 10% inside. So the solution that the cell is in has a higher concentration, which means that it is hypertonic. So now, once again, what way does water move? Water moves from where there is a lot of water to a little water. So there is more water inside the cell than outside of the cell, 90% to 80%. So my water is going to go from an area of high water to low water, which means that my water is going to move out in order to help me reach equilibrium. So water will move out of the cell. All right, one last example. I'm sure you have an idea of which one this will be, but let's go through it anyway. So picture looks the same. 10% solute means 90% water. 10% solute means 90% water. So if we're compare the inside or the outside of the cell to the inside, they are the same, which means that my solution is isotonic. Now, one thing to keep in mind with isotonic, a lot of the times we think of that the concentration of the solute in the water is equal, but that's not true. Those two numbers don't have to be equal. What you're looking at is the solute concentration outside and the solute concentration inside. Okay, so make sure that you're able to distinguish between those two things. Now, if it's isotonic, that means that we've already reached equilibrium. But if you recall from some of our previous work, just because we're at equilibrium does not mean that molecules stop moving. So what's going to happen is we are going to have water move into and out of our cell. So water will move in and out of the cell. So I'm going to put up some examples. I want you to try them. Check your answers on your Ed puzzle, and then we can go from there. All right, here's your answer for question 1. And practice question 2. Give this one a shot. Check your answers. And here are your answers for question 2. And go ahead and give question number 3 a shot. And here's your answers for question number 3. So 2 more things I want to talk about is how these different tonicities of solutions will affect different cells in different ways. So first we want to look at animal cells. So in a hypotonic solution, the animal cell will burst or it will lyse. So that's our first picture down here at the bottom on the left. So with all that water rushing in, because a animal cell does not have a cell wall to provide it with that really rigid structure, it can't withhold all that water. So think of it as a balloon. Eventually, if you continue to fill a balloon with water, it's going to eventually burst. The same thing can happen with animal cells. Next, in a hypertonic solution, so this is your picture on the very far right, animal cells will start to shrivel up and shrink. So because so much water is moving out, the cells are losing water, which causes them to shrink down. Also not a really good thing for the cells in the animal. In isotonic solutions, that's our picture in the middle right there. It's described as normal. There's not too much water, there's not too little water. So the isotonic solution is ideal for animal cells. So animal cells ideally are in an isotonic solution. So they're not gaining too much water or losing too much water as a result of osmosis. Plant cells are going to be a little bit different. So in a hypertonic solution, that's your one on the very far left here, your animal cells will start to wilt or be plasmolized. And in this situation, if you've ever seen a plant that looks like super droopy, you automatically know, oh, that needs water. Well, it needs water because the plant cells have shriveled up, and that vacuole that stores the water has released as much water as it can, because in that hypertonic solution, water is moving out of the cell. In your hypotonic solution, that's our one on the very far right here, the vacuole is filled up with water, filling. But because we have a cell wall that is different than the animal cell, that cell wall can withstand all of that pressure as it continues to push out against it. So you can see kind of how that cell in your picture here is, it's kind of pushing out against that cell wall. It's puffing up. And that's called turgid. So that's when you see a plant that is, it's straight up, the leaves are firm. This is our ideal solution for plants. So they're storing water. That water makes those plant cells nice and big and firm. That's an ideal situation for a plant. Now, not as bad as in a hypertonic, but in an isotonic solution, the vacuole isn't filled completely. And not filling that vacuole up completely makes it so we're not putting that pressure against the cell wall, and our cells, not our cells, plant cells, haven't puffed up as much. So they're kind of floppy, they're flaccid. So this is a plant where you can see it's a little bit droopy, but the leaves haven't started to shrivel up completely. So just kind of having an idea of how animal cells and plant cells are going to react differently to these different solutions. So that is everything. Take a look at your coloring packets and the practice that you have to do with tonicity and the movement of water.